Um, the topic today, my topic is nanotips and microtips for DNA extraction and disease diagnosis. Basically, the platform using a small tips, that's the platform that my group is developing. And the concept is very simple. We are going to use a very tiny tip to concentrate a very low concentration of molecules to a small terminal end of tip area, and then we want to detect it, what kind of species or what kind of biomarkers are there. So overall, in my lab, I'm doing like three kinds of work. The first one is micro scale and nanoscale fabrication, so manufacturing. So I'm designing like small structures, and then on the basis of this small structure, I'm assembling molecules, especially using an electric field and fluid flow and other chemical bindings. And using those structures, I'm applying for sensors and actuators. And recently, in order to use these platforms in a field, I'm working on a small scale fuel cell with a high power density. So the topics today is I'm going to introduce a nanotips from the fabrication through the physics based on a capillary action and electric field, and also viscosity. And then based on the working principle, I want to show you how the concentrated DNA can be expressed in bioinstrument. And the second topic is a microtip. It's a little bit like a larger scale because the target is larger than a DNA. So microtips, we are working on this micro scale tips. This one is a microtips for low cost and then point of care diagnosis of tuberculosis. So let me go with the first topic. So the concentration of molecules is really important. For instance, molecules within the range of 1 to 100 nanometer, it starts from a protein molecules, and also viral particles, they are about 50 nanometer, 100 nanometer, and 150 nanometers. A little bit larger particles, they are bacteria. Okay? And those small molecules, what kind of molecules are there, especially with low concentration? That's really of great interest in the field of medicine and biology and environmental technology and monitoring. So many of approaches have been developed. The first method is centrifugation. We can use the inertia to concentrate small mass of particles, but as you expect, when the particle size is below 100 nanometer, the inertia is very negligible. So it needs a very long and then cumbersome steps to concentrate molecules. And microfiltration, we can use filters having like a small pores to filtrate a particles. However, this step again is very cumbersome, and also it creates a lot of shear stress on the particles, so they are very quickly damaged. And the third one is the most recent method. So they are using a magnetic particles. So magnetic particles, they are bound with target molecules, and then we can concentrate them using a magnet. Okay? However, the efficiency is not such a good for this magnetic method. So if you see a centrifugation method, then we can put a blood sample or human sample, and then after centrifuge, we can see the cells at the bottom and molecules at the top. So we can use those supernatant to concentrate a particles, but we need this high speed, 100,000 G gravity, to concentrate a molecules for 10 hours or 20 hours. Is really time consuming, and then it needs a lot of efforts. And the microfiltration is like a very simple. So in a paper or some of polymer, it has a small pores. And then when we put a human samples on top, then some of particles can be bound, and then rest of them, they can be filtered out. However, the process is very inefficient and then cumbersome. So also it needs a lot of concentration steps with a centrifuge or other methods. And then also the molecules can be very easily damaged during the process. And this approach is readily recent. So this tube, we have a target, the magnetic particles, and then we put samples. Then target molecules, they are bound onto a large magnetic particles. Okay. Then after incubation of, for example, 1 hour, we can use a magnet to attract these magnetic particles. So if you use a magnet, then they will be concentrated then at the bottom. And then we can remove this dirty solution. It can be like blood or human other samples. Then in order to detect these targets, we can add up a pure solution. Then actually, the solution amount was like this big, and then the solution amount was reduced by about 1/3. So concentration efficiency is only like a few times, but it was purified in the process. So the purification can be increased a little bit, but efficiency of concentration is not that good. And recently, electric field has been used to concentrate particles. We can manipulate an electric field to control the particles. We can concentrate them or release them in combination with microfluidic devices. So similar type of concentration devices has been studied a lot previously. If you see this kind of electrode, then we have a circular shape of electrode with a ring. Then when we apply electric potential, then it was found that DNA can be aggregated on top of this top electrode with a electro-osmotic flow. And also 20 nanometer, 10 nanometer, and then 5 nanometer particles, they can be attracted between two electrodes. So two electrodes, they are colored with dark, and then small dots, they are gold particles. So somehow those particles can be attracted between two electrodes. However, still the electric field strength is not enough on planar electrodes. So instead, I want to try a very tiny tip. Then the advantage is at the tip end, electric field strength will be very high. Then it can generate more force to attract the particles. So this nanotip, a nanotip was fabricated, and then it was immersed in a solution. And then in order to have some of pressure, the solution was hang in a metal coil. And then electric field was applied. Then we expect to see a capillary action around the nanotip, and then based on the electric field, particle will be attracted onto the surface of a nanotip. Okay, and then we will withdraw the tip out of the solution. Then this capillary action that makes some of filtration function, because as you see, if particle is here, if particle diameter is comparable or smaller than this tip diameter, then it will be left due to the capillary action to hold this particle and then pull this particle. However, if this particle is greater than the tip diameter, like this big, then it will be pulled down to the solution. okay? Because the capillary action to pull down this particle is larger than the capillary action holding this particle. So we can have many advantages with this platform. For example, concentration using a high electric field strength is possible through this. So if we use planar electrodes, then we can generate about 10 to the power of 5 volt per meter. But using a small tip, we can make 10 to the power of 7 volt per meter only with 10 volt. Okay? And also, we can collect particles depending on a size. So we do not need any of a centrifuge or microfiltration. We just put the tip in and then apply field and withdraw. Then automatically we can collect the particles depending on their size. And also we can coat this nanotip with specific molecules. Then, for example, we have viral particles or other proteins. Then they will be bound on top of that. And then when we withdraw non-specifically bound particles, they will be removed, or we can rinse them out. And only the specific molecules, they can be collected through this nanotip. And we can use fluorescence detection by adding up fluorescence particles in order to identify what kind of particles are there. And one of the advantages is to use this electrical detection. So by having a small tip end, we can significantly reduce the electrical double layer effect on the tip surface. So we can reduce a lot of capacitance effect. Then we can measure a true resistance depending on the particles on the surface. Then after having the particle onto these nanotips, we can use a lot of other analysis tool to identify these particles. And in this presentation, I just want to show you these 2 and these 3, total 5 functions. Okay, so in there, what's missing is a specific detection. And the specific detection will be shown through a microtip detection. So I want to shortly discuss about a dielectrophoresis. That's a kind of one of our main mechanisms. So if we apply an electric field, then the electric field strength at the tip is 10 to the power of 7, 10 million volt per meter. It's really high strength of a field. Like you can imagine, our height is, for example, 1 meter. Then if we apply 10 million volt for us, then everything will be gone, right? So such a high field is created at the end of a tip. And if we have a target particle in the solution, it will be attracted. So the attraction force is proportional to the volume of a particle, because that will be coupled with the polarization length of a particle. And then we have a constant with the gradient of electric field. So this, the main contribution of this nanotip is to increase the field strength. Then that will proportionally increase the force to attract concentrate particles. And in this graph, that shows scaling issues. So particle size is from 1 nanometer and then 10 nanometer, 100 nanometer, and then 1 micron, 10 micron. So our target is within this range. And Brownian motion, it will be negligible by having a larger particle. And it will be crossed with dielectrophoresis force. Then at a certain point, dielectrophoretic force will be greater than this Brownian motion. By using a nanotips, we can make it shift the graph over dielectrophoresis higher. Then we can attract the particles smaller than 10 nanometer up to 1 nanometer, okay, close to 1 nanometer. And this is a simple configuration for a experimental setup. So we have a solution, and then in the solution we have a nanoparticles, and then we want to use a nanotip to concentrate a particles. Then basically we can think of two forces. One is a dielectrophoretic force that will attract particles using an electric field. Okay, that's a positive force. And then on the other direction, this Brownian motion force, thermally induced force, will be random. Then we can compare these two forces. Then particles in there that will be attracted by an electric field, but also random motion will inhibit the attraction. So we can draw this line. Then within this volume, the dielectrophoretic force will be greater than Brownian motion. Then particles in this volume will be attracted. However, the particles out there, it will not be attracted because the Brownian motion will be dominant. So our process becomes random. Through the computation, using a supercomputer, all the computation was done by a Liu group at Northwestern University. We made a very small tip in there, and then this size is about like a 2 millimeter, okay? And the tip diameter is from 50 nanometer through 5,000, like a 5 micron. So we changed the tip diameter and then also changed the particle diameter. So particle diameter was changed from 1 nanometer to 10 nanometer and then 100 nanometer. So in the simulation, what you want to know is how the effective distance will be changed according to the particle diameter. So if our target is 1 nanometer, then we will follow this curve. What I mean is distance from the tip end, it's a 1 micron, then we can generate this amount, like 0.1 piconewton of force. And with the increase of the particle diameter, we will have a larger force. So it will be increased with the increase of the particle diameter that can be compared with the Brownian motion induced force. So this line that shows a Brownian motion induced force according to the particle diameter. Then at 1 nanometer, we follow up this line, and then with 100 nanometer, we follow up this line. Then when we compare the Brownian motion induced force with dielectrophoretic force for 100 nanometer, they do not meet each other, right? That means a particle over 100 nanometer that will be always attracted to a tip. However, when you see a 1 nanometer particle, then they will meet at 20 micron. So what I mean is a particle within 20 micron distance that will be attracted. Otherwise, the particles out of 20 micron from the tip surface that will not be attracted. So that will be dominated by the random motion. So we learned what kind of particles in terms of diameters they will be attracted to the tip surface. And then we fabricated nanotips. Looks like that. So it's kind of looks not such a clean, but it works really well. Okay? So we have small tips, and then tip end we have a silicon carbide nanowire. The diameter is about 500 nanometer, and it's coated with singular carbon nanotubes in order to make these silicon carbide nanowires electrically conductive. So we have these branched dendritic structures, and then at the tip end, we can concentrate the particles. Of course, the electric field will be generated on the dendritic structures. So particles also will be attracted around that range. This movie shows the attraction procedure. So we have a small solution. We see only the top meniscus due to the scale. And then we will dip this nanowire into a solution. And then we will wait about like 1 minute until the particles are attracted. And then after the attraction, we will withdraw this tip out of the solution. then particles will be attached, bound onto the surface by the non-specific binding. And when we withdraw the tip, of course, the capillary action, that was really dominant, so that will filter out large particles in the solution. And we checked for gold nanoparticles, because gold nanoparticles, their properties are well defined. So we used those gold nanoparticles to compare our experimental result with the computational result. Then we tested this for 2 nanometer and 10 nanometer and 100 nanometer particles with the various concentrations. And then this is the end of a tip, and then this part, that's the end of a silicon carbide nanowire. And if you see the dimension, then it's about like a 6 nanometer. Then the width shown here is about 200 nanometer. And we can detect gold nanoparticles. Its diameter is about a 2 nanometer. So these black dots, they are the gold nanoparticles. And in this way, we could see how many, what can be the sensitivity. In a limited volume, we have 100 million particles or million particles. Then what is the detection limit to see the particles? So here, x-axis, that shows a gold nanoparticle diameter, and then y-axis shows the limit of detection. So with increase of nanoparticle diameter, of course, we will see the increase of the detection limit, because a larger particle will experience a larger force. So according to our calculation, that's the black dots. We showed this straight line in log scale. And also, when we do the experiment, they were kind of closely matched each other. So red dots, they are the experiment. The interesting point that we discovered in the experiment was this tip was made of like a lot of branch structures that created a lot of singularity points for electric field. So that could enhance the concentration performance. So according to our calculation, we got this curve. So instead of a single-ended tip, when we use the dendritic nanotips, the detection limit was reduced a lot, increased a lot by order of 6 orders of magnitude. Okay? It was a really remarkable change. And then, however, you know, when we increase the diameter, we will have a less gain. For example, 2 or 3 orders of magnitude, because dielectric forces is so dominant. Whether we use a single tip or dendritic tip, they will show a kind of a similar result. And then also we have done the experiment and then checked. They follow up quite well in the nanoscale concentration experiment. And then using the tip, we want to see what kind of physical phenomena we can generate. So we have a nanotip, and then now we want to use this nanotip to collect a specific particle in a human sample. For example, blood or saliva. But you know, if you see saliva, then it has a lot of bacterial particles and cells and DNA and proteins, a lot of other debris, fibers. So we want to collect a small solution, a small particle that is smaller than a tip diameter. And we want to know what kind of physics is involved. Then, conceptually, when we concentrate particles using an electric field, then large particles and small particles will be attracted. But depending on the size, large particles will be more likely attracted. And then, when we withdraw the tips out of a solution, a meniscus will be formed like that. This film is coupled with a viscosity. So this viscous film will form, and then that can capture the particles on the surface. And then second thing is the capillary action. So now, when we withdraw this tip completely, then splitting of a solution will happen at the point. Then if the splitting happens exactly at the center of the particle, then this particle is at a point. It can be captured or pulled down to the solution. If this splitting point goes up, then the capillary action from the bottom that will be a larger than this withdrawal force. Then particle will be dropped. So those kind of capillary induced a size specific concentration was studied. So this is a rough mathematical model, and then we calculated a capillary force induced on this particle, and also we calculated a force for viscosity. And in experiment, what we found was on a nanotip surface. If, for example, diameter over this tip is 360 nanometer, then we could capture up to 125 nanometer diameter. Then aspect ratio is a 0.35. That can be, that explains, compared with the tip diameter, 0.3 times of particles of this tip diameter, those particles can be captured. And then we repeated a lot of these experiments, and then we found that we cannot capture the particles over 700 or 950 nanometer diameter particles when a tip diameter is about a 500 nanometer range. And then we have drawn this graph. Okay? So on the x-axis, that's the nanosphere diameter. So we tested 100 nanometer and then 300 and multiple diameters of nanoparticles. And then also we measured a force. Also with the analytical equation, we could find that capillary force pull down the particle to the solution. It was calculated with this one. Okay? And also dielectrophoretic force to holding this particle to the tip surface, that was calculated with this blue curve. And also the viscous force that's holding this particle onto the tip surface, that was also measured. Okay? That's the green point. So the blue and then green, they are the attractive force to the tip. And then this one is the force to pull down the particle out of the tip surface. So when we add up these two, then those two forces, combined forces, reach the red point. And at a certain point, due to the small force from the viscosity, we cannot attract the particles anymore. So that was around a 600 nanometer. That was consistent with the experimental result. Okay. Then we want to use that principle to concentrate a DNA. So the diameter of DNA is about like a nanometer range. So once we attract DNA, then we can concentrate them and then attach them onto the surface. However, the cells or other bacterial cells, they will be removed from the surface. So we have a solution with the DNA and then see whether we can attract particles or not. So we put the tip in the solution, and after 1 minute, we have withdrawn the tip out of the solution. Okay. Then we can see the nanotip up to this white spot, and then after that, you will see a very tiny, like a fiber, diameter of about like a 500 nanometer. That's the fiber made of DNA. So also, you know, we can use another tools like a fluorescence microscope or electron microscope to confirm that's the DNA out of the solution. And we compared two cases. One is the DNA in solution with the cells, okay? And then the other is the DNA without any of the cells. So we compare those two curves, and then interestingly, depending on the concentration, we could see a similar graph. So we increased the concentration of DNA, and then it was attracted in pure solution only with the DNA. But even when there are particles in the solution, the same amount of DNA was captured onto the tip surface. Okay? Yeah. So those size selectivity happened through the nanotips, and of course, we want to go for more of actual application. So what we do here is we have a tube, and then in the tube we can put a saliva in there, and in our saliva we have a large amount of human DNA. So now it takes about 1 hour to purify the DNA through the commercial kits. Okay? But it's really time consuming, and then we need about 45 steps, okay, to purify the DNA from saliva. So if I ask my students do the experiment, purify the DNA with the commercial kits for 8 samples, then 8 multiplied by 45, right? Then so many steps, then always they are making a mistake. But using this nanotip, it can finish up the concentration purification with a single step. So we prepare a nanotip, and then here is a saliva, including a human DNA in there, and then put the tip in the solution and wait about 1 minute, and then withdraw. Then DNA will be captured, and then that was studied. The number of DNA was counted by a amplification tool, real-time PCR. So through this way, we could concentrate a DNA out of the solution. So at the beginning, we spiked a lambda DNA, okay, that we know the sequence. So microgram and 10 nanogram and this kind of concentration. And then y-axis, that shows the threshold cycle. This one is a kind of a log scale of number of copies of DNA. And then small number means we have larger amount. So it's kind of inverse. So when we see the performance of nanotips, at certain concentration, we could detect this many using a nanotips. And also, when we use commercial kit, that's expressed with a triangle, then we could get almost the same yield of DNA, but with just like a single step concentration. So from saliva, it worked really well to concentrate and purify the DNA. So what this means is you spit on the floor, okay, then I can tell your DNA within 1 minute. Okay? So now we are working on this nanotips, and also it was applied to detect DNA in our saliva, human DNA. Then the yield was 19, and then it was equivalent to commercial kit, but with a very simple and then low-cost method. So in summary, we studied the dielectrophoretic concentration using a nanotip in experiment and then numerical simulation, and they agreed quite well. Through the size selective concentration using dielectrophoresis and capillary action induced force and viscosity, we could directly concentrate DNA from a sample mixture. And based on this working principle, we could directly extract human genomic DNA from a saliva. So that's the non-specific concentration of molecules from human samples. And now what I want to discuss is a microtips. It's a little bit like a larger scale because bacteria is greater than DNA, so we can use larger tools. Okay? And then these microtips mainly developed for low cost and point of care diagnosis of tuberculosis. So I think as you are aware of this tuberculosis, it's a worldwide, it's a deadly disease. So if you see the map, then red area is area that the TB is really prevalent. So the China and then South Africa and North Korea, but now not South Korea. Okay, South Korea, I think it's a sky blue. Okay, similar to Brazil, and also in Brazil, relatively like a large population of tuberculosis. The thing is, about 1/3 population, okay, of world population, one third of them, they are already infected with this tuberculosis. So when our immune system is down, then at certain point, the cells will be grown in our body, and then we are kind of significantly affected by those TB. And about 14 million active cases in a year. It's a really large number, and over 1 million deaths per year. So a lot of efforts are being made to rapidly diagnose this TB. Okay? Mm-hmm. But the problem is still the tools to detect this TB is problematic. So if you see methods, then mycobacterial culture is one of the gold standards. So we can culture the cells in sputum. Okay? Then we know whether a person has a TB or not. The problem is it takes about 4 weeks, 1 month. Okay? So it's too slow. And smear microscopy is to count the cells under the microscope. So they are stained, and then we can count how many cells. But it's not sensitive or it's not specific. Okay? So it's not such a reliable tool. And then one of the most reliable tools is detection of DNA. However, its cost is high, and then still it's very slow. And also skin test, it's not sensitive at all. And chest radiography, specificity and sensitivity is not that high. So the challenge was defined through this way. So we need a very rapid test within 30 minutes. That means we want to tell a patient that disease can be, you have a TB or not within 30 minutes. That means in a single visit. Otherwise, patient will come to a hospital and then give the sample and it's gone. Then we don't know where a patient is gone, right? So that was a kind of big problem. So we want to make it point of care. And then second one is a very big challenge. It's low cost. Okay? Now, if we pay about $100, then we know this person has a TB or not. The problem is, in South Africa, okay, for 1 week, one kid can survive with about $3 to $4. They can buy a bread with that. Okay, that's better than spending dollars for diagnosis. Okay, so low cost means we want to make it within $2 or $3, and sensitivity, specificity should be high. So we want to make it low cost, but we do not want to compromise the performance. So that's the challenge that we have. So technically, the challenge is, large sample volume means 1 milliliter, then roughly we can make a size like 26 millimeter and 10 millimeter and 4 millimeter. And in this sample volume, the size of bacteria is 1 micrometer. Okay? 1 out of 10,000. So we cannot see the cells out there directly. Then, if we zoom in, the number of cells is only 10 or 100. Okay? It's really challenging to concentrate those. And even that comes with other cells that we do not want to detect. About 40,000 species, then we need to detect 1 cell out of a few million bacteria. Okay? That's the challenge. And then one more challenge is the medium is highly viscous. Okay? So that's the reason why this problem is not solved for the previous 100 years. So there have been many methods for the microfiltration and using a magnetic beads and also microfluidic devices, but they have problems in the sensitivity and specificity or a cost. So current methods do not meet the demands for a point of care diagnosis of TB. So for this purpose, we developed a method to concentrate particles in a multiscale. Since the particle size is small and then their number is small with a large volume, we try to concentrate them using mechanical flow. So we shake the solution, then particle will be circulated. Then those particles will be delivered within a few millimeter range. Then as far as they are going there, then we can use an AC electric field to concentrate further those particles to the surface. And then tip surface is coated with antibodies that specifically recognize whether this cell is a TB or not. If they are not TB, then they are flown away. So this multi-scale concentration mechanism was developed to concentrate bacteria. So in order to make a 30 minutes boundary constraint, we developed a method to dissipate a sputum for 15 minutes. And then we are concentrating a particle for 2 minutes plus some more preparation. And then detection was done with a fluorescence microscope using a fluorescence antibodies. So for this purpose, we fabricated a small tips. Okay? So in one wafer, we can make about 400 chips. Okay? And each chip has 5 microtips. And then this chip is installed in a coupon. And then the whole thing is disposable. Okay? And if we see the tip end, then it has some of a wiggled shape to generate a large electric field. So this one shows a device operation. So we have a small tip installed, and whole device, the size is the size of this water bottle. Okay? Then after setting up the tool, it will dive into the solution very gently. Then for 2 minutes, the cells will be concentrated, and then it will come out of the solution. So if you see the this operation procedure, then this simple procedure that has multiple physics to concentrate target cells onto the tips. And this one shows the overall mechanism. So in a large well, like a 20 millimeter and then 10 millimeter, in this scale, we want to circulate the cells by shaking the whole solution. And then particles will be further concentrated within 100 or 200 micron range by using an electric field. And then they will be, some of particles will be bound. Of course, there will be a non-specifically bound cells. And when we withdraw the tips out of the solution, these particles will be dropped due to the capillary action. Then we have only the target cells and label it with the fluorescence. Then we know we can detect this bacteria. So concentration mechanism was validated through the measurement and also through the simulation. And also we can see that the particles, they are attached onto the tip surface. Those are the target bacteria using an electric field. So they attract cells onto the surface. Okay? And we checked how many cells they are attracted. Often the combination of electric field with flow, and then only flow, and then electric field, and then without any other mechanism. Then we see that larger number of cells, they are attracted when we combine electric field and then streaming flow, mechanical flow. And the tip was coated with antibodies and then target cells, they will be attached on top. Then we can use a fluorescence antibody to identify the target cells. And then we check the specificity. So we have a target bacteria with 100 particles per milliliter. And then non-specific, other like bacteria, E. coli or Staph epidermidis bacteria with 10,000 concentration. But still, the signal is lower than a target. Okay? So specificity was quite good. And we went for a sensitivity test. So as you see, it was kind of increased with the increase of the concentration of BCG. So we can concentrate up to 10 CFU, colony forming unit per milliliter. And we spiked a bacteria in sputum, and then we found that we can see the signal from 100 CFU per mil. So it was a quite good sensitivity and specificity. And as you see on the tip surface, they were attracted onto the tip surface and then captured. That is expressed with white spots. And at the detection limit of 200 CFU per mil, we tested about like 40 times. Out of them, 12 is negative control. We have just like a sputum, don't have target cells. But this one is target cells over 200 CFU per mil in 1 milliliter of sputum. Then success rate was quite good. It was 96% over yield at the detection limit. So when we compare our system with the gene expert system that is very recently developed, biomarker, this one is a DNA, but ours is a bacteria and then DNA, also using a nanotips. And limit of detection was same. And then in terms of cost, the current system is quite expensive. It's not applicable for low-income countries. And then our platform is about like $1,500. And then assay cost, we are trying to make it like a few dollars. But in US, I think it will be expensive. That's not my decision. And also, you know, power requirement, 100 or more than 100 watt is necessary. But you know, if you go to like South Africa, then electricity is on and off. Okay, so this kind of large power is not available with a continuous base reliability. Okay, so ours it was designed to make it with batteries, and then assay time we can make 25 minutes versus 2 hours. Still, we have to increase the throughput, and also we have to detect a DNA out there. So this is a kind of a recent prototype that we are working on. So in the device, we plug in the well. This well is a small well that can contain 5 microliter. And then 4 tips, they are assembled on this coupon. Also with a magnet, after loading the solution, this coupon will be loaded in the device. So in the sample, we have a DNA in saliva, and attach this coupon with a magnet. Then if we press this button, then it goes into that, and then after 1 minute, it's withdrawn. Then we can detect those targets. And this one is a more simplified version for electrical measurement. And for this whole work, you know, I'm working with a very kind of big group in like a medical school, and also Seattle Biomedical Research, and also other labs within mechanical engineering department. And also NSF and Washington research grant, and also Center for Disease Control and Prevention, and NIH, and Catalyst Foundation with the Bill and Melinda Gates Foundation, and also manufacturer for the prototype device. And I appreciate Professor Duda for making this travel possible. Okay? Yeah. Thanks for your attention. Okay. Yeah. Thank you. Heating. Mm-hmm. Yes. Yeah, it can be possible, but as you know, the size of tips are very small, so it can heat up a very small solution, like a microliters. Yeah, that's the kind of problem. But if it meets the need, then yeah, sure, it can be used. Right. Okay. Yeah, that's a good question. So. When we use a high-frequency electric field, it does not attract just like simply charged particles. So when we use a DC field, then the field will attract all the charged particles, then the tip surface is contaminated. So by using a AC field, we can generate a dielectrophoresis, then we can attract only the polarized particles. So that will give more of selectivity in terms of particles. Mm-hmm. Yeah, they will be removed. However, when we use a DC field, for example, too many particles, they are attracted. For example, including fibers in like blood, there are a lot of like particles, including proteins, and then also platelets and other particles. Once they are, all of them, they are attracted, then tip is like coated with a very thick layer. Then none of those size selective function does not work because the tip is already contaminated in the solution. So when we use a very high frequency, we can choose the particles with a more selection, but that was not discussed in detail in this presentation. That's right, yeah. Mm-hmm. Exactly. Yeah. You mean the device? Okay. Now you know we completed the semi-clinical test, and then there are two versions of the device. So the first version was a non-specific concentration. So we do not need a decision for medical purpose. So the device is kind of under manufacturing. So commercial device will be available within a year or two with sales distribution. And then for this diagnostic device, we just finished the semi-clinical test in the last year and plan for the clinical test. So after finishing up, then we can, we know the reliability of the device. Then with more optimization, we expect about 6 years, including FDA approval. Okay. Mm-hmm. Yes. Yes, antibodies and then tip size, and then well size. So if we increase the volume, then we can contain more targets in there. So it's more advantageous. But for small targets like a protein, then we have to use nanotips, small tips, and then small solution. Then still we can have advantage over sensitivity. Yeah. Okay. Okay, yeah. Thanks for your attention again. Thank you.